Hi, everybody. My name is Liang Huang from Baidu Research USA and Oregon State University. I'm very glad to share with you our work on better mRNA vaccine design using computational linguistics algorithms. And this is a joint work with between Baidu Research and Stamina on Therapeutics, which is a vaccine company in Shanghai mostly, and also includes the collaboration with University of Rochester and Oregon State University. So we all know that mRNA vaccine from BioNTech, Pfizer, and Moderna has been a huge success to fight the pandemic. But there's one challenge that still remains, that is the chemical instability of mRNAs. And as a result, the relatively low immunogenicity. And as we all know, RNAs are mostly single-stranded, and as a result, they can be easily degraded or cut down into pieces. So that's low stability. And as a result, you can produce less proteins because you have less full-length or intact mRNAs. Only the full-length mRNAs are useful to produce the full spike protein, the real spike protein. And in turn, you have less antibody response because you have less proteins to trigger them. So how do you deal with that? Well, you need ultra low temperatures for storage and equipment, which is a problem for some countries in the world, and you need larger doses. But that would trigger more side effects. We all know that almost everybody around me got very bad fever after the second dose of mRNA vaccines. So that's probably due to the fact that they have relatively larger doses. So our goal is to design better mRNA sequences using computational algorithms, which will yield more proteins and as a result, yield more antibody response. So here's the mathematical problem of mRNA design. You want to design a sequence that will translate to the spike protein. And the spike protein has 1,273 amino acids and plus a stop codon. But each amino acid can be translated by multiple triplet codons. So that's the famous redundancy in our genetic code. And we're going to exploit the fact that they have redundancy in our genetic code so that you have multiple choices per amino acid. So for example, valine has 4, leucine has 6. And you list the choices, number of choices, and you multiply them all together. You got a humongous number, 2.4 times 10 to the power 633. That is the number of valid mRNA vaccine sequences. Each one of them is a good candidate for vaccine design. But which one is the best, right? So Moderna's recent paper in PNAS showed that the more stable, the better, because the more stable mRNA would stay longer and will yield more proteins, which is what we talked about in the previous slide. So your question is, how do you find the most stable sequence among all these alternatives? This exponentially large search space, right? That's extremely hard because this number is much, much more than the number of atoms in the universe. And if you want to just look at the sequences 1 second at a time, you will need much longer than the life of the universe to finish that task. So you definitely cannot do enumeration or exhaustive search. So how do you do more efficient search? So very luckily we designed a very efficient dynamic programming algorithm that is a generalization of classical RNA folding algorithm for a single sequence to multiple sequences. And then you can solve this question in less than 10 minutes. So that's very fast. So let's look at the result. So here's wild type in the virus. That's the virus's original sequence to encode spike protein. And as a result, you can see that it's very, very unstable. You got tons of single-stranded regions, which can be cut down to pieces. After about 10 minutes, less than 10 minutes of our design, you got a very different sequence and a very different structure, right? Because you used very different codons. But both sequences translate to the same spike protein, right? So in other words, they are synonymous sequences, but they have very different sequences and very different structures. On the right, you got even like almost a DNA kind of purely double-stranded structure, right? And has a much lower free energy than the wild type. And this is computational estimation of the stability, right? And so here's the wild type. Here's the most stable sequence of structure. But that's only one of the 2 dimensions. That's the x-axis, the free energy. You want the lower free energy, the better, or the most stable possible. But that's only one of the 2 dimensions. The other dimension we're going to consider is the translation efficiency. So Moderna's paper also showed that you would want to prefer frequent codons that human cells would like to use and avoid rare codons. It's well known that, for example, leucine, you would prefer some codon like CTG 41% of the time and do not use TTA at all, right? And each amino acid has a distribution over its codons. So they're not equal. You want to prefer the frequent ones as much as possible. So that's the y-axis, which is called CAI, or codon adaptation index, right? But that's very easy to optimize. Unlike energy, energy is very hard to optimize, but the CAI is so easy, so you can just use greedy algorithms. So in fact, that's the traditional codon optimization technique used by most of the vaccine companies, including BioNTech and Moderna. So their sequences, as we know today, only use codon optimization. So they're very, very close to the CI greedy design, which is always use the best codon for each amino acid, right? So between our most stable design and the CI greedy design is the feasibility limit curve, where you can change the hyperparameter lambda, which is the relative importance of CAI. So if lambda is 0, you only consider energy and you got the most stable, the lowest free energy design. But if you increase the lambda, gradually you reach the CI greedy point where you only consider CI and not energy, right? And this curve is the feasibility limit in the sense that beyond that, you have no feasible no region or design at all, because we want to go top left as much as possible. But underneath that curve is the feasibility region, and you have a huge number of candidates, right? But so far to this day, most vaccine companies only explored this area, which is the code optimization, without knowing that there's a huge area that is uncharted or unexplored, because there's no algorithm to go to this point, or that point previously, right? So there's no way to kind of go beyond this line. Now, our work showed that this is a huge area that needs to be explored, and we're gonna explore them not just in silico, but also in vitro and in vivo in the next few slides. So this is work done by Stomi Therapeutics, a vaccine company in Shanghai, and they took several of our designs plus their baseline sequence, which is basically codon optimization, very similar to Moderna's BioNTech's, and then we compare, say, stability. And in solution stability up to day 5, you can see that our most stable design A, the lowest free energy one among our designs, stay always on top, and the baseline H is the least stable, and at day 4 it runs out. It means it has 0% that is intact. Right? This is the percentage of full length or intact mRNAs at the end of day 1, day 2, and day whatever, day 4. And if you just look at day 4, yeah, our A has 35%, and G being much worse in energy has only 16%, and H, the baseline, has 0%. That's very well matching with our theory. But more importantly is the immunogenicity or the antibody response. That's the thing that we care about in vaccine design, right? So here we use 6 mice per design, and at day 0 and day 14, you got 2 shots, the first shot and the booster shot. And after another 14 days, you measure the blood sample. This is very similar to our human injection of mRNA vaccines. And we measure the spike protein specific neutralizing antibody titration. So that's the standard way of measuring antibody response level. You can see that baseline H is basically useless. It doesn't trigger much antibody response at all. But our designs, especially the more stable ones, A, B, C, D, got a lot better, right? Very, very significantly better. And in particular, the C design here has almost 30-fold more antibody response than the baseline. So that's very encouraging. So with that, I would like to thank you for your attention.